Okay, if everyone is ready, let us start. Assalamualaikum warahmatullahi wabarakatuh and a very good morning everyone. Dan salam kekeluargaan. My name is Ahmad from Pusat Kepakaran Subfertility, Lembaga Penduduk dan Pembangunan Keluarga Negara. I will be the moderator for today's session. Ladies and gentlemen, welcome to our 5th series of International Scientific Webinar 2025. We are going to have an interesting topic this morning entitled X-linked Mysteries, Unravel Genetic. Before we begin, we would like to share some housekeeping announcement to ensure our session this morning will run smoothly. Number one, our webinar session today is being recorded. The recording will be made available on our official YouTube channel, Persatuan Pegawai Sains Malaysia. Number two, the link for today's attendance and participant certificate will be shared in the chat box 15 minutes after we begin. Number three, you can share the questions to the speaker in the chat box too. We will try to answer all the questions at the end of the presentation. Ladies and gentlemen, it is our pleasure to introduce our today's speaker. She is a dedicated science officer at the Pathology Department of Hospital Tunku Azizah, Kuala Lumpur. Graduated with bachelor degree in genetic and molecular biology from the University of Malaya. She also earned her PhD in genomic and biosciences from the University of Bath in the UK. With a strong passion for diagnostics, research, and teaching, she brings a wealth of knowledge to the field. Her special interests lie in rare diseases and imprinting disorders, area in which she continued to contribute with curiosity and commitment. Now, everyone, please welcome Dr. Sasi Devi Saminathan. Over to you, Dr. Sasi.
Thank you very much, Mr. Muhammad, for the introduction. Very good morning, everybody, ladies and gentlemen, and salam sejahtera. Thank you. I would like to thank Persatuan Pegawai Sains Malaysia for giving me this opportunity to present to you regarding this genetic topic. So without much ado, I'd like to start the presentation. So everybody might be wondering, what is this X-linked mysteries? So this is actually a type of genetic inheritance, and we will be unraveling the genetic secrets as we go along. So the presentation has been, will be presented in such a way that the mysteries will be incorporated with the secrets, the genetic secrets. So as means of introduction for X-linked mysteries, X-linked inheritance is actually a pattern where the gene responsible for a trait or disorder is located on the X chromosome. So as we all know, males have got the X and the Y chromosome as their sex chromosomes, and the females have got 2 X chromosomes as their sex chromosomes. So in total, humans have got 23 pairs of chromosomes.
Slide presentation. Doctor hasn't shared the slide presentation.
I beg your pardon, sorry about that. Can you see my presentation now? Okay, sorry about that. So let's carry on. So I haven't moved on in terms of the slides yet. I'm still talking on this the topic, the title slide. So we were at the males have got X and Y chromosome, and the females have got 2 X chromosome. So in total, there are 46 chromosomes in a human being. Okay, so or we say it as 23 pairs of chromosome. So 22 pairs are known as the autosomes, okay, or autosomal chromosomes, and 1 pair, which is either the XY in the males or the XX in the females, are known as the sex chromosome. So today, the entire talk is going to be about this sex chromosome, particularly the X chromosome. Okay, so what are the mysteries and the secrets? So why do some female carriers show symptoms when they carry the X's? And why no male-to-male transmission happens with X-linked inheritance? And what explains variable severity in affected males? So all these answers are actually in the genetics of the person and much to do with X inactivation, a mechanism called X inactivation, mosaicism, genetic complexity. We will be visiting these terms as we go along, some of these terms, okay? So the basic genetic concept that we have to understand is, first of all, the DNA structure. So what is the DNA structure? The double helix structure made out of? It is made of 4 nucleotides, which is the A, T, C, and G. So this DNA is found within the chromosomes that I explained earlier. And this ATCG forms the basic unit of inheritance. And the genes are carried on chromosomes, which are long strands of DNA. So the role of X chromosome is that the females have got 2 X chromosomes and males have 1, as I explained earlier. And the X chromosome itself contains over 1,000 genes related to various functions, including development, immunity, and reproduction. There is a mechanism called X inactivation that happens in females because females have got 2 X chromosomes. So one of the X chromosome, is randomly inactivated in each cells in order to balance the gene expression between sexes. Okay, so this also ensures that the females do not have double the dosage of X-linked genes. So now the basics of X-linked inheritance, okay? When inheritance has to do with the X chromosome. So the genes that each of the condition will be referring to the genes on the X chromosome. And so in males, because they only have 1 X chromosome, if a mutation is present in that X chromosome, then the mutation is expressed and they will be affected. Okay? But whereas because the females have got 2 X's, if only 1 X is affected, so they become carriers, okay? And they only become affected for a certain condition or disorder if both of their X's are mutated. So in this type of inheritance, we do not see male-to-male transmission. It always goes from male to a carrier and then it is transmitted, okay? So now we go a little bit deeper into the X-linked genetics. So this is an inheritance pattern where the genetic responsible for a trait or disorder is located in the X chromosomes. Okay, we already know that males have got XY and they only have 1 single copy of the X, and that one is enough to express the trait or disorder. Whereas females, because they've got 2 copies, they actually need 1 copy from each parent in order to express the trait or disorder. So why is this X-linked genetics so important, right? Many significant genetic disorders are actually X-linked, especially those conditions that affect males. So studying X-linked inheritance helps us understand the mechanism behind genetic diseases. Okay, so now X-linked inheritance, there are 2 types. So one is called the X-linked recessive inheritance, and the other one's called the X-linked dominant. Okay, let's look at it one by one. So the X-linked recessive inheritance, it affects the males. Okay, affected males, XY, they inherit the mutated gene from their mother through her X chromosome, and the females, XX, must inherit the mutation from both parents in order to express the disorder. Or carrier females, heterozygous, that means they have a combination, they're not homozygous, they don't have the same alleles, okay? So these carrier females do not show symptoms, but they can pass the mutated gene to their offsprings. There is also a ratio in which they pass on this mutated gene. So examples of diseases that are X-linked recessive, for instance, hemophilia and Duchenne muscular dystrophy. And in terms of the pattern, the sons of a carrier female have got 50% chance of inheriting the affected X and expressing the disease. Daughters of the carrier female have 50% chance of being carriers themselves. So this is a pedigree chart. So a round filled circle is an affected female, and a square unfilled is an unaffected male. So when an affected female has children with an unaffected male, then she produces children who are unaffected, the white ones, as well as children who are affected. Okay, so and then and in this case, where the children, both of them are affected, they produce all children, both male and females, who are affected. Whereas in this case, you'd see that this daughter of hers doesn't seem to be affected, but when she has her own children, she has one affected male. This is because she is a carrier of the condition that is inherited through an X-linked recessive trait. Okay, so in this X-linked recessive inheritance, males are relatively more affected than females, and the genetic trait never skips a generation. There is always someone affected in each generation, and only the affected mother can transfer the disease to the male individuals, and disease transmission from a father to a son will not occur. Okay, so this is another diagram to show, to make it look, to show a more clearer visual for you. So this is the unaffected father with the X and Y chromosome, mother with double X, yeah, and she has children, an affected son, because she's passed on her X chromosome to this son, so he is affected. She has also passed on her affected X chromosome to her daughter, but because the daughter has got two X chromosomes, she is only a carrier. And in this case, she passed on her normal X chromosome to her unaffected son, and similarly, her normal X chromosome to her unaffected daughter. Right? So in the case of an affected father and an unaffected mother, so the father passes on his X chromosome definitely to the daughters. So both his daughters are carrier daughters, okay? Because they also receive another normal copy from the mother, so they don't themselves express the disorder. Whereas in the case of the sons, because the father only passes down the Y chromosome to the son, so the sons will not be affected by the condition. Okay, so a few carriers may be mildly affected due to skewed X inactivation, which we will come to later on. Okay, so that was X-linked recessive inheritance pattern. Now we look at X-linked dominant inheritance pattern. Okay, so in a dominant inheritance pattern, only one copy of the mutated gene on the X chromosome is enough to cause the disorder, whether it's inherited from the mother or the father. In recessive, we need both to be affected. So males XY with the mutation often show more severe symptoms due to having only one X chromosomes. Females XX who inherit the mutation may exhibit milder symptoms as they have a second normal X chromosome. Okay, the example of diseases that are inherited in an X-linked dominant inheritance pattern is Fragile X syndrome and Rett syndrome. So the pattern is the affected males will pass the disorder to all their daughters, but not to their sons, and affected females can pass the disorder to both daughters and sons. So this is a pedigree chart, the same concept. If it is filled, they are affected, and if it's clear, they are not affected. So in this case, both males and females are likely to be affected because the mother is passing on her X-linked X chromosome to all her children. Yeah, so they are likely to be affected. But she could also pass on her normal chromosome in these cases. Okay, so the genetic traits may skip a generation in X-linked dominant. In X-linked recessive, it does not. And the affected father can transfer the disease to all female individuals. And male-to-male disease transmission does not occur even in X-linked dominant. Okay, this is the same diagram, a different diagram, but showing X-linked dominance. So in this case, the affected mother has got her affected X chromosome there. So she passes it on to her daughter and her son, and both of them are affected. In the case of X-linked recessive, the daughter will only be a carrier, but in this case, they are both affected. So when she passes on her normal X chromosome to the daughter and the son, they are unaffected. Okay, so when the father is the affected one, he is passing on his X chromosome only to his daughters. So all his daughters will be affected, but his sons will not be affected. So I hope that gives you an idea about the difference between X-linked recessive and X-linked dominant. So we've talked about X-linked recessive and X-linked dominant, but just to let you know that there are also other forms of inheritance patterns. For example, I spoke about autosomes just now. All the chromosomes apart from the sex chromosomes, they also are divided into autosomal dominant inheritance as well as autosomal recessive. And there is also another type of inheritance, which is Y-linked. This is specifically to do with the Y chromosome. So in terms of the mechanism of expression, certain things can happen that skews or changes the expression. So one of it is called the lionization or random X inactivation. Okay, so because the females have got two X's, they can be randomly inactivated. And also there are skewed inactivation. So in skewed inactivation, we can find sometimes symptomatic females, although they should be carriers, but they can be symptomatic females. But the degree to which the disorder is seen in them may be milder. So it could also be because of X deletions or uniparental disomy. This is where you get both copies of the gene from the same parent. That's called uniparental disomy, or some kind of structural changes that affects the phenotype. Okay, so a little bit further, talking more about X inactivation and skewed X inactivation. In females, one chromosome is inactivated, but this process isn't always random. Sometimes skewed X inactivation leads to one X chromosome being predominantly active in all cells, which may result in disease expression despite having a healthy X chromosome. Okay, so now the mystery, right? Because we're talking about mystery and the secrets. So how does skewed X inactivation occur, and how does it influence the severity of symptoms in females? We are going to look at it as we go on, okay? So, and also when you have gene mutation in X-linked disorders, for example, mutation in genes like the dystrophin, it will lead to progressive muscle weakness, right? So the secret is the specific gene mutation in the X chromosome can disrupt critical proteins involved in muscle function, blood clotting, and neurological development. Okay. So now that was just generally talking about it. So now we look more into the mysteries, okay? I think I don't need to repeat this again to you because this has been covered already. So the mystery now, why do some females show symptoms of X-linked diseases when they typically have two chromosomes, including one normal one? Why do they show it, right? So what's the secret? It's to do with these two mechanisms. One is called the X inactivation, yeah, and the other one is called the skewed X inactivation. So in females, one of the two chromosomes in each cell is randomly inactivated. I showed you earlier. This means that most genes located on the X chromosome, only one copy, either the maternal or the paternal X, is expressed in each cell. So typically, this process ensures that females don't double the gene dosage for X-linked genes compared to males. Okay, so this is an important mechanism that compensate for that is involved in making in why some females show symptoms of X-linked diseases. So there's the other mechanism is called skewed X inactivation. In some cases, X inactivation can be skewed. It doesn't happen normally. Okay, so meaning one X chromosome is preferentially inactivated in a larger proportion of cells than the other. And if the mutated X chromosome is the one that remains active in a majority of cells, a female will manifest symptoms of the X-linked disorder, even if she's a carrier. A carrier means they usually won't show symptom, but in this case, she will, right? So this is particularly relevant in conditions like Duchenne muscular dystrophy that we will be looking at it, more into later, and hemophilia, which is also another X-linked recessive disorder, where the mutation on the X chromosome causes disease symptoms when the healthy X is inactivated too much. Okay, so we already know that these disorders have come up quite a bit now from the first slide. So just a quick, how to say, a mystery and the secret about these diseases, okay? So affected males will show symptom in hemophilia, while females often act as carrier without symptoms. So what's the mystery? Why do some female carriers of hemophilia develop symptoms, right? So the secret is some female carriers of hemophilia, they develop symptoms because of that skewed X inactivation, okay? In these cases, the X chromosome carrying the normal gene is preferentially inactivated in most cells, and then leaving the X chromosome with the hemophilia mutation active. As a result, the female expresses the disease similar to affected males. Okay, that was hemophilia. So now in Duchenne muscular dystrophy, this is a problem with the dystrophin gene. It has a mutation that leads to muscle degeneration in males, with females often having milder forms of the disorder due to the second X chromosome. Okay, so now the mystery is, why do females with the same mutation as the males often have milder symptoms than males? Okay, so the secret is because the females with the same mutation often have, because they have two X chromosomes. So the normal X chromosome can compensate for the mutated one. So in contrast, males only have one X chromosome. So a mutation on their single X results in full expression of the disorder. Okay, there's another disorder that I threw in here, which is the Fragile X syndrome. This is an intellectual disability and developmental delays caused by a mutation in a gene known as FMR1. So the mystery here is, why does a mutation on the X chromosome affect males so severely, while females typically exhibit milder symptoms? This is going to go back to the same secrets, but bear with me. So the secret is, a mutation on the X chromosome affects males severely because they have only one X chromosome. If that X carries a mutation, there is no backup copy, leading to full expression of the disorder. Females, on the other hand, have two chromosomes, so the normal X can often compensate for the mutated ones, resulting in milder symptoms. Okay, so now we come into one particular, just talking about Duchenne muscular dystrophy. We're going to look at it a little bit more in detail. So DMD is very severe, and it has an early onset, and it's a progressive disease. Okay. On the other hand, the same variant of the disease, but a milder one, is called the Becker muscular dystrophy, and it has a later onset. And DMD gene, which is mutated in this condition, is the largest human gene. It has 79 exons. Okay. And the deletions in the gene, the hotspots, are usually between exon 45 to 55, and it involves large deletions of exons. Okay, so the diagnosis that's used for these conditions usually CK, creatine kinase, enzyme test, MLPA. I'll go into it more in detail later. NGS is next generation sequencing. It's usually used for precision medicine. Okay. So this is a graphic about how the patient looks like and the conditions. So in terms of hemophilia A and B, so there is a mutation in the F8 gene on the X chromosome, and is usually severe bleeding episodes, joint bleeding, and hematrosis, bleeding into joints, especially after minor trauma or surgery. And it's more common than hemophilia B, and affects 1 in 5,000 male births. So B Similar to A, with spontaneous bleeding episodes and joint damage. It's less common though, affecting 1 in 25,000 male birth. Okay, so both types are X-linked recessive and typically affecting males. Deficiency of clotting factor leads to impaired blood clotting and prolonged bleeding. And in terms of the treatment for this disease, the factor replacement therapy or regular infusion of the deficient clotting factor is in place. And also gene therapy, promising research in gene therapy aims to provide long-term relief for this condition. So usually hemophilia A and B has got complications, joint damage, and potential for life-threatening bleeds, okay, leading to arthritis as well. So this, I just showed you two types of X-linked disorders, okay? So now let's move on. There are other X-linked recessive disorders. To name a few, G6PD deficiency, it leads to hemolytic anemia, especially after oxidative stress. And there are risk factors, triggers, and screening methods. XLA, because caused by no antibodies. Less Nihan, due to uric acid and neurobehavioral issues. Then there is ALD, Fabry, Menkes, and Hunter. There's also red-green color blindness, okay? This is inability to differentiate red and green colors. This is genetic, yet, and it is an X-linked recessive disorder, okay? So now, in terms of X-linked recessive disorder, we want to look closely at this condition, dystrophy, Duchenne muscular dystrophy, and Becker muscular dystrophy. So these are the clinical manifestation. You can read it. I do not want to read the whole thing. But basically, the onset is between age 3 to 6 years. It's a progressive weakness, and patient usually is presented with pseudohypertrophy of the calf muscles and the spinal deformity and cardiopulmonary involvement. Okay, so there'll be mild to moderate MR as well. So usually this is a test that we do in our lab in Hospital Tunku Azizah. So usually when the referrals come in, they will either have GOAS positive sign, or they will have a pseudo hypertrophy of calves, and they will have an elevated CK level, and so on. Okay, so there is a section later on to see the clinical manifestations and clinical symptoms of DMD. So DMD is caused by the mutation in the dystrophin gene, where in the X chromosome, it is X-linked recessive, we already know. So it affects males mostly, and the affected males get their mutated gene from their mother, and females are usually carriers, okay? So the symptom begins between 2 to 5 years old, and then the individual will usually lose the ability to walk by age 12. And the life expectancy is typically in the late 20s to early 30s. Although now advancements in care are extending this lifespan. So the genetic mechanism is dystrophin, a protein essential for muscle function, is missing or defective in individuals with DMD, leading to muscle degeneration. Okay, so males are more commonly affected. Same pedigree, I'll show the pedigree later, but it's the same as the X-linked recessive pedigree, okay? And so, yes, the dystrophin, a protein essential for muscle function, is missing and leading to muscle degeneration. So now, what are the current treatment options for people with DMD? No cure, but steroid treatments such as prednisone can help slow down muscle degeneration, which will lengthen their life a little bit. So the gene therapy and exon skipping to bypass the defective part of the gene are areas of active research at the moment. So one thing that exon skipping therapy can do is that it can skip the exon, which can cause a more severe phenotype like DMD and make it into a milder phenotype, which is BMD. Okay, so that's one of the advantages of exon skipping therapy. So now this is a pedigree chart. Same concept applies. If it is filled, it's affected. If it's a circle, it is a carrier. So this is a carrier mother married to a normal father, and she has one affected son and one carrier daughter. So this affected son married a normal female, and the children are one female carrier and 2 affected DMD males. Okay, and in terms of this normal son male who married a carrier female, the carrier produced a carrier daughter and 2 normal sons and 1 affected male. Okay, so this is the pedigree chart. It's very similar to the X-linked recessive chart that I showed you. Then now let's move on. So what are the clinical symptoms for referral for this condition? So they usually have early motor delays. Okay, they can't sit, they have delays in the milestones for sitting, walking, crawling, and they have difficulties in activities that requires muscle strength, standing up, sitting, climbing stairs, all that. So there's progressive muscle weakness, usually in the proximal muscles, muscles closest to the trunk, such as the hips, thighs, and shoulders. There's difficulty walking or frequent falls, and the ghost sign, a characteristic method of rising from the floor where the child uses their hand to walk up, their legs to stand, excuse me. So there's also noticeable muscle wasting, and there's calf hypertrophy, which is the enlargement of the calf muscles due to muscle tissues being replaced by fat and scar tissues. Okay, so there's delayed developmental growth, there's cardiac and respiratory issues with DMD patients, heart problems, especially dilated cardiomyopathy, difficulties in breathing, and so on. So the criteria for referral is a child with progressive muscle weakness, difficulty walking, and delayed motor milestone. This patient can be referred. So the genetic testing for DMD is indicated, particularly if dystrophin deficiency is suspected. So what do we do when we receive their samples? So this is the molecular genetic testing strategy for Duchenne muscular dystrophy. So let's say the clinical indication is for testing DMD and all the clinical signs like progressive muscle weakness and Gowers sign, calf hypertrophy is there. Then this testing is particularly important when there is also a male child with muscle weakness and the family history of muscular dystrophy. So it's very important to do a family testing as well. Okay, so the initial genetic testing approach would be, because DMD is mostly at least 70% of it, 65 to 75% of it is caused by large scale deletions. Okay, large deletions, that means it deletes not just one nucleotide, AGTC, but it deletes the entire exon. So MLPA, or multiplex ligation dependent probe amplification, becomes a dosage analysis, becomes the primary method for detecting this disease. Okay, so that's the first line method, and then we use that method to detect large deletions and duplications. And MLPA is highly sensitive and can identify deletions, duplications in the entire dystrophin gene. Okay, especially this method is especially useful for detecting mutations that involve multiple exons. And as we know, the dystrophin gene is a very huge gene, so it's a very useful technique to use that first to test. Okay, and MLPA can detect 65 to 70% of the DMD cases. Okay, so what happens to the 30% that is not detected by, which is not caused by a large deletion or duplication? So that is possibly caused by point mutation, which is mutation in single base pair changes, okay, like in one of the nucleotide A, G, C, or T. Okay, so that could be in any one of the genes within the 79 exons. Okay, so a point mutation analysis of the entire exons, 79 exons, has to be done in order to determine whether they are actually normal in terms of, like, not having the disease. So so just a point to add here, because it's such a huge gene and because it's very tedious to do sequencing for each one of the exons like that, so latest advancements like the next generation sequencing allows us to more quickly identify if there is any point mutations within the dystrophin gene. Okay, so we also do confirmatory testing, confirmatory diagnostic testing. Once a mutation is found, you can do a confirmation with an array CGH or different molecular methods, okay? And we also do a carrier testing. So once a male DMD patient is already identified in the family, the mother has to be tested if she is a carrier, first of all. And then all the female relatives will also be tested. And if there is symptomatic male siblings, they will also be offered the test, okay? If assumingly we have done the MLPA, we have done the sequencing, and still there is no solution to that case, yeah, the odyssey goes on, then we have to consider that to test for other differential diagnosis of DMDs, okay? So test for other muscular dystrophies, okay? Okay, that's about it. So that was X-linked recessive. Now let's look more closely into X-linked dominant disorders. So these are 4 disorders that I picked. One is Rett syndrome. Why I picked that is because we do it in our laboratory, and this is caused by mutation in the MACP2 gene, and it primarily affects females due to male typically not surviving to birth. And the symptoms of Rett syndrome is loss of motor skills, speech, stereotypical hand movements, and development regression after normal early development. There's also Fragile X syndrome. This is due to an expansion of the CGG repeat in the FMR1 gene, and the symptoms intellectual disability, hyperactivity, repetitive behaviors, and developmental delays. And it is more severe in males due to having only 1 X chromosome. Okay, and also there is incontinentia pigmenti. This is caused by a mutation in IKPKG gene. The symptoms is rash at birth, followed by Blasco lines. I wanted to show you a picture of that, but unfortunately I didn't manage it. So it involves the skins, the central nervous system, eyes, and teeth. Also, there's hypophosphatemic rickets. The cause is a mutation in FAX gene, leading to a phosphate wasting in the kidneys, okay? And the symptoms, rickets, bone deformities, and growth problems. And the treatment for this, at least there's a treatment. So phosphate supplements and vitamin D can be given to improve bone health. So now we look specifically at case study of Rett syndrome. So these are 2 girls who are affected by, Rett syndrome. These are images that I got from online. Yeah. So we know now that Rett syndrome is caused by a mutation in the MACP2 gene on the X chromosome. Okay, it is an X-linked dominant inheritance, and although both males and females can inherit the disorder, it primarily affects females, as males with mutation typically do not survive to birth or early infancy. Most of them die, so we don't even see them, okay? Most of the cases that we see are the females. And the incidence is approximately 1 in 10,000 live female births. And the symptoms and progression, the early symptoms are between 6 to 18 months. The development seems normal for the first 6 to 18 months, after which the child may lose acquired skills such as speech and purposeful hand movements. So the main features are loss of purposeful hand movement. Example, they can't grasp, they can't reach, yeah. Then there is stereotypical hand movements like ringing and clapping, and slowed growth, motor abnormalities, example, gait problems, and severe cognitive impairment. And other symptoms will be seizures, breathing irregularities, and autistic behavior. So MACP2 gene, the function is it's responsible for producing a protein that regulates the expression of other genes in the brain. Okay, so mutation in the MACP2 prevents normal brain development, leading to the loss of motor and cognitive skills. So why is it that it affects females more severely? Because males typically have only 1 X chromosome, and a mutation in MACP2 results in severe symptoms or death early in life. Okay, so actually what happens is, it's not that it doesn't affect the males, but because they die anyway, so the females are the ones who are more severely affected. Okay, so females having 2 chromosomes may have a normal 2nd X chromosome that compensates for the defective one in some cells, which is why they can survive but often experience significant development regression. Okay, this is a pedigree chart. Although this is actually for all these conditions, yeah, incontinentia pigmenti, Rett syndrome, as well as vitamin D. So if you see, so this is an affected male, yeah, somehow he has children, and there are all the females are affected because they are getting an X chromosome from the father. And you only need one copy of the X in order to get the condition. So that in this situation also, because the mother could have passed on the normal X's to these other children, so only 2 of the children are affected. Okay, so this is the X-linked dominant chart, and it continues on to the next generation as well, right? So affected females pass the mutation to 50% of their daughters and sons. That is X-linked dominant, yeah? Affected males pass the mutation to all their daughters, but none of their sons. Okay, so in this case, you see all his daughters affected. Okay, and where else? And in this case too, because he's an affected male, all his daughters are affected, and in this case as well. Okay, that is X-linked dominant pedigree. So now we have an overview of the Rett syndrome, more detail. So currently no cure exists, but treatment focuses on managing symptoms and improving quality of life. So there are therapies, physical therapy and speech therapy. They help with motor function and communication. And there are medications for seizures and behavior issues, but there's no proper complete cure, okay? And then there are ongoing researches into gene therapy and drug treatments that is aiming to target that MACP2 gene or the proteins involved, okay? So in terms of the mutation, so the first line genetic test for Rett syndrome is sequencing the MACP2 gene, okay? This is the MACP2 gene. So when it is mutated or deleted, then it causes the problem, okay? So MACP2 or methyl CpG binding protein 2 is the gene responsible for Rett syndrome. Mutation in these genes prevent normal brain development and function. Common mutations include point mutation. It could either be substitution, insertion, or deletion in the gene sequence, with the R113C mutation being one of the most common. Okay, so if you do sequencing, you'll find that quite a lot. So what are the clinical symptoms for referral for this condition? So normal early development followed by regression. First, they develop fine, 6 to 18 months, then they regress, okay? So they lose especially speech and purposeful hand movements. And then they also lose the motor control and interest in social interaction. So basically a little bit more like autistic behavior, yeah? And then there's the abnormal hand movements, stereotypical hand movements. They always wring their hands, clapping, repetitive hand-to-mouth motions, and so on. These hand movements are often noticed around the time of skill regression. Then there is cognitive and speech regression as well, and severe intellectual disability. Okay, so there's difficulty with communicating, including a loss of social interaction skills and many repetitive behaviors. So they also have ataxia, unsteady movement, gait abnormalities, and muscle weakness. They have loss of purposeful hand movement. So this is motor abnormalities. Then they also have autonomic dysfunction and seizures. The seizures will begin between age 1 to 4, and then the breathing irregularities, including hyperventilation, hypoventilation, all that happens during sleep. There's growth retardation or abnormal weight gain, okay? And there is abnormality in the facial feature. I had one picture of a girl with, I didn't want to put it because it was a little bit extreme. So it's kind of like her entire jaws and her lips are like sort of missing. So they have prominent jaw, large ears, and long face, which may become apparent as the child grows, okay? Facial abnormalities. And usually they are referred due to developmental regression after normal early development, as I said earlier on. And then if Rett syndrome is suspected, the first genetic testing will be to look at MACP2 gene mutations, okay? Especially if females are showing this regression and hand-wringing behavior. That's classic symptom. So what is a molecular genetic testing strategy for Rett syndrome? So if Rett syndrome is suspected, these are the clinical indications we already went through, so I'm not going to repeat it. So the first line test for Rett syndrome is sequencing. Why? Because 80 to 90% of the condition is caused by some form of a mutation in the MACP2 gene. Okay, so Sains sequencing is the most common method used for MACP2 testing, and identifying point mutations in the gene. Also, nowadays, with the advancement of the sequencing, we could do next generation sequencing, where panels can also be used to test for MACP2 mutations. And we can make a panel along with other genes associated with developmental delay or autism spectrum disorders. Also, there is this 8 to 10% of Rett syndrome, which is caused by large deletions or duplications in the MACP2 gene. So we can do the same technique, MLPA, for Rett syndrome as well. So it will at least detect 8 to 10% of the cases. Okay, so MLPA is sensitive to detect large-scale structural changes in MACP2 genes that can contribute to Rett syndrome. Okay, so structural in the sense like when a huge deletion happens, like when an exon is deleted, that's huge. Yeah. So this is useful in cases where Sanger sequencing shows no point mutation, but clinical symptoms suggest Rett syndrome. So that will be our second line testing, MLPA. Okay, so summary in terms of molecular genetic testing, both of these tests we are doing in our laboratory. So the first line testing for MLPA is for large deletions, duplications in the dystrophin gene. The second line testing, sequencing of the dystrophin gene for point mutations, MLPA is negative. And then we also do some confirmatory tests in cases that require it, okay? In terms of Rett syndrome, the first line testing is MACP2 gene sequencing for point mutation. And the second line testing is MLPA for deletion duplications in the MACP2 gene, and also confirmatory testing MACP2 to confirm the diagnosis of Rett syndrome. So carrier testing and inactivation studies may also be useful for genetic counseling and understanding the clinical presentation. So this applies to both Duchenne as well as Rett syndrome. So in terms of treatment and research for all these disorders that I've been talking about. So there is gene therapy, and it's happening a lot in DMD and hemophilia. So for DMD, it will be exon skipping, exon skipping therapy. So as I explained earlier, it will skip an exon that will otherwise produce a more severe outcome. So when it skips, so it rescues the reading frame, we call it, yeah? So when it rescues the reading frame, we will call it an in-frame mutation that will cause a milder form of it, and that's what's called Becker muscular dystrophy. So there's also newer technologies like CRISPR and newborn screening programs. All of them are advancing, and there is potential for treatment for these diseases. So more specifically for CRISPR-Cas9. Okay, now we look at it from the same theme of the presentation. Like, so what is the mystery, right? The genetic mystery about gene therapy targeting X-linked disorders. How can we successfully use these cutting-edge techniques to deliver genetic material precisely? Excuse me. Where it is needed, and how do we ensure long-term efficacy and safety? So the answer to that is successfully using cutting-edge technique like gene therapy and CRISPR-Cas9 involves ensuring that genetic material is delivered to the correct cells with precision. Okay, so this requires targeted delivery systems, which is the CRISPR-Cas9. Okay, excuse me, I'm not feeling so well. Such as viral vectors or nanoparticles. So we use that and we direct the therapy to the right tissue. So long-term efficacy and safety are ensured through extensive clinical trials, monitoring for off-target effects, and ongoing adjustments to treatment protocols. So this is the secret, how we successfully use these sort of cutting-edge techniques. Otherwise, it all sounds very bombastic. This is really realistic, but it is, okay? So in terms of the skewed X inactivation in research, there are ongoing research they're investigating how to correct or predict skew X inactivation patterns, okay? And their role in the manifestation of X-linked disease. So when it becomes skewed, it interrupts the usual manifestations, right? So the mystery is, how can we predict and correct these patterns to better understand disease development and improve therapies? So the secret is it can be achieved through combination of genetic testing and advanced computational methods, okay, that analyze X inactivation patterns in various tissues. So techniques like gene expression profiling and single-cell RNA sequencing, these are all very high-end techniques, okay, in molecular genetics, can help identify which X chromosome is more active. So to improve therapies, targeted interventions such as gene editing. Gene editing means you use CRISPR-Cas now. And epigenetic modulation can help restore balance, X inactivation, or compensate for dysfunctional genes. Okay, all these can lead to more effective treatments. So what are the challenges and ethical consideration? There is a dilemma, okay? So the genetic testing for X-linked disorders can be controversial, particularly regarding prenatal testing and the decision to test minors, okay? So how do we balance this? How do we balance the benefits of early diagnosis and treatment with the potential risk of social and ethical consequences? So it involves emotional and psychological impact on the patient and family, and privacy of genetic information, and the potential for discrimination. So whoever's handling these have to handle it with care. Okay? So while early detection can lead to better management and outcome, it also raises ethical concerns about informed consent, then the right to know, and the possible societal stigma. Therefore, careful genetic counseling is essential to navigate these challenges. So in all of these genetic conditions, rare disorders, we will always encourage genetic counseling because with genetic counseling, the affected, the parents of the affected children can make more better informed choices, as well as the extended family of an affected patient. Okay, so it's important also for family planning, genetic counseling, okay, carrier testing, pedigree analysis, and all that. So prenatal and pre-implantation diagnosis is also recommended, especially for ladies who have affected males with X-linked recessive disorders, for example, like DMD. Yeah, so prenatal pre-implantation is very important. So also ethical and psychological, psychosocial support. Okay, so this is also important to consider. So, what is the summary in terms of the X-linked mysteries and unraveling genetic secrets? Well, the mysteries that we have looked at is why is it that some female carriers show symptoms despite having only one normal X chromosome? And why is it that there is no male-to-male transmission? If the father has it, why doesn't his son get it, right? Specifically of the X-linked diseases. And what explains the variable severity in affected males, okay? Why males are more affected and why it is variable. So the secret lies in the X chromosome mechanisms, okay? So X inactivation and skewed X inactivation that we already looked at. In females, only one chromosome is randomly inactivated, but sometimes skewed inactivation can lead to disease expression despite a normal X chromosome. And then there's this other concept that I just touched upon. It's called mosaicism. So mosaicism is the presence of two distinct populations of cells, okay? So one with a mutated gene and one without. So one normal, one mutated. And that contributes to variable disease expression, okay? And genetic complexity. So mutation in X-linked genes disrupt critical proteins involved in muscle function, neurological development, and other essential processes leading to disease. So advances is understanding the treatment. So there are many ongoing research in CRISPR, Cas9, yeah, gene editing technologies, gene therapy, and also exon skipping therapy, which was specifically for the DMD disorder, okay? All these technologies promises to address the underlying genetic cause and offer potential cures for X-linked disorders. So ethical consideration of genetic testing, early diagnosis and treatment is also required for careful balance. Okay, so we looked at the unique X-linked transmission patterns between recessive X-linked recessive and X-linked dominant inheritance, and also we studied some pedigree charts. Okay, and also we learned that female expressions are variable because of the nature of X chromosome, okay, and X inactivation as well as skewed X inactivation. So that brings us to the end of my presentation. I just have to make a disclaimer. So I've made every attempt to acknowledge the source of graphics used in this presentation. These are not my own works, just like, pictures that I got online, or stock photos online, and I don't take credit for them. Okay, thank you. Any questions?
Thank you so much, Dr. Sasi. It was a very wonderful presentation from you. And right now, I believe all participants have a few knowledge, or at least all the secrets and mystery have all been unraveled by now. So let us proceed with the Q&A session. With me, I have a few questions from the floor, from the participants. Let us go through one by one. The first one is, just now you have mentioned a few genetic testing available in HTA Kuala Lumpur. They are asking whether university hospital or private hospital can send a referral cases to your lab.
Currently, the testing that is related to X-linked disorder that we have in our lab is Duchenne muscular dystrophy and Becker muscular dystrophy, which is the similar test, but we look at two conditions, related conditions, and also Rett syndrome. So in terms of answering whether or not universities can send samples, first of all, any genetic cases will have to go through the clinical genetic team. So they will have to review your cases. And then you have to contact the head of the laboratory in HTA, who's Dr. Chin Loy Kim. And then upon her approval, then you can proceed to send samples to the lab. Yeah. So that's the procedure as far as I know. Yeah.
Let us move on to the next question. Is there any study regarding the prevalence of X-linked disorders in Malaysia? And maybe I want to add into this question is there any relation for this X-linked disorder with the consanguineous marriage or the closely related marriage? Or is there any relationship that for marriage between interracial marriage can help to better prevent this X-linked disorder?
Yeah. So I'll just repeat. I caught the part about consanguineous marriage, whether it will how to say affect the outcome. What was the first part? Sorry.
The prevalence. Is there any study regarding the
about the
prevalence of the disorders, X-linked disorders?
To my knowledge, we still have to check with Institute of Medical Research, where they do a lot of research, genetic research as well. But from our lab, we are looking into studying the Duchenne muscular dystrophy in more detail. So we want to look at the prevalence in Malaysia, and we also want to look at the different types of mutations that we are finding within our population, because mostly the reference that we have are references of mutations found in other countries, like the Western countries, and some Asian results are also out these days. But it will be very good for us to have our own data set, like Malaysian data set. So we are working on that. So for Duchenne muscular dystrophy, yes, and also we are currently optimizing neuromuscular panel. So some of, if let's say it has not tested as a DMD, then we can possibly proceed to the neuromuscular panel to see if there is any other differential diagnosis that fits with the condition. In terms of the consanguineous parents, yes, it does affect the expression pattern. I had a point somewhere made about it, which I can't find quite in my note at the moment. But I don't mind elaborating more through maybe email later on the consanguineous.
Sure.
Yeah.
So basically, the final question we got here about the challenge that you face in the lab for this genetic testing, or in general in your lab. aside from the ethical challenge that you mentioned before, is there any other challenges that you'd like to share for us to, you know?
In terms of the, if let's say we talk about Duchenne muscular dystrophy testing, right? So the sheer size of the gene is a challenge already. So for many years now, we've been doing the first line of testing, which is MLPA, right? So and it detects about 70% of the cases. Anyway, so but there are still all these normal cases we can't actually say which tested normal for MLPA. We can't actually say that they are normal for BMD. Definitely, you know. So in our reports, we always have to say that further testing has to be done to account for that 30% of the cases that are caused by point mutation. So I believe that can be solved with higher end technologies like next generation sequencing, whereupon you can look at the entire all the coding regions of the gene, and it will be a little less labor intensive. Otherwise, to do sequencing for the entire 79 exons of dystrophin gene is going to require a lot of manpower, time, and also cost. So yeah, the basic problem that most labs will face is manpower, budget, and also the time frame that is required. Because this is a diagnostic lab, we don't have the luxury of spending so long on a case. We have to have to release results within the required turnaround time. I hope that answers the question. Yeah.
Thank you so much, Dr. Sasi. I'm afraid that's all the time that we have right now. So thank you everyone for your participation. Let us meet again on the next series. Thank you, everyone. And they're asking for your slide. Would you share your slide to us?
Yes, I would. Yeah, no problem. I will send it to you. Yeah. And if there are any questions, I don't mind taking it over the email later on. Yeah. Thank you.
Okay. Thank you so much, everyone.
Thank you so much for the opportunity.